mute your audio, and number two, introduce yourself to everybody by just via chat, just type in a little bit about yourself. And we're gradually getting more people herded into the proper location, and then we'll get started, because we have a fair amount of material to cover today.
So why don't you go ahead and begin while I do this?
So should we?
One. You go ahead. I'm going to need to step out and do this elsewhere so that I don't interfere.
Okay. 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 As I was saying, take time to just type in on the chat window if you figured that out yet with the Zoom software. Just type in something to all of us to tell us who you are. Doesn't have to be anything too formal. We're just delaying it a couple minutes before we get everybody set up, and we'll start with our discussion shortly.
I would go ahead since it's 5 after.
Okay.
Go ahead.
Okay. So I guess we're going to get started now. In case you don't know, I'm Roger Barthelson. I'm coming to you live from the Keating Building on the University of Arizona campus in Tucson, Arizona, where it'll probably be in the upper 70s today, in case you're freezing to death wherever you are. And my engineer who's assisting me is Martha Narrow. And our focus for our discussion today will be next-gen sequencing, getting started, but especially read cleanup, which requires also read evaluation. And let's get into that. Now, many of you are often tempted to ignore this, and you've just gotten your data from the core facility, and you're excited to see whether you're seeing changes in expression, whether how well you can assemble a genome or a transcriptome, or all of the above. But and so you're tempted to jump in and start using whatever tool that you've heard of or you have experience with, and not do some of the most important steps of your whole analysis, which is to say the cleanup step. Because chances are, during the sequencing process, stray bits of information have entered your data, and some of that is just plain junk. And as you probably heard elsewhere, if you have junk to start with, you're going to end up with junk in the end. And most of your data may be perfectly clean. But when you're talking about assembling a transcriptome or assembling a genome, it's very hard to put those overlaps together, however it's done, and do it accurately if you don't first remove any stray information, because otherwise your software may be trying to match up primers or adapter sequences to each other, And maybe they'll do that, but it won't really help you in terms of understanding the biology in the long run. So one of the ways I think that it's really easy and good to approach doing cleanup is this pipeline of software that I've put together in the past year. And that's, of course, my personal prejudice, but I think it helps you go through in a fairly formalized manner to assemble your data in an intelligent fashion. It forces you to ask questions of yourself and understand your data a little more in some cases. And it helps you analyze the quality and prepare to do the cleanup steps, the trimming steps that are important for downstream processing. And the apps in the HTProcess pipeline, and this is sort of a broad view of the pipeline, which is not necessary today, but it helps you understand why things are set up this way, is that HTProcess is designed to help you move larger batches of information through the discovery environment, through the analyses, without having to run a lot of individual jobs in each read or read file. And it also helps store information and carry that through the analyses so that you don't have to keep track as much of each read name and the downstream files that are generated from that read file, and remember or have to look up what are the specific properties of that file so that you can figure everything out. The information is assessed in the beginning, it's stored as much as possible, and then the information is used in the downstream applications for doing data analysis.
Give me a minute.
So the question may come up, well, since we have all these apps that are designed for Tuxedo pipeline, is this only for RNA-Seq? And the answer is no. HTProcess apps are not just for RNA-Seq. They're also for genome analysis or genome assembly, for example. And so eventually we will be building HTProcess apps specifically for genome assembly, and to some extent we have already. But for now, the cleanup tools are good, handy tools for everybody. As I was saying before, it's a pipeline. So the starting app is the HTProcess prepare directories and run FastQC, which is a mouthful, but that's what it does. It prepares the directories for entry of the data in an intelligent fashion. I mean intelligent in the sense that it separates your reads into the separate pairs, the forward or the first read file with its paired second read file or reverse read file, and also the single read files if those are involved in the beginning. And you don't have to have information for each of the boxes, but you do if you're going to enter paired reads, then you do have to put something in the 1 box and the 2 box, and that just makes sense. But part of the read cleanup process is that the information is gathered by prepared directories and run FastQC, and then you're ready to run HTProcess Trim-O-Matic after you've actually interpreted the data. And as I said, it goes through, helps you bring in the data. And once you have, and once you've filled those boxes with the reads as you have them. And I should also say at this point that it's important to consider how you're going to use this set of tools. I've designed it so that you're dealing with the read library. And this may be annoying to some people because they may just feel they just want to put all the reads in one box and that's it. But chances are, if you're doing a larger project, not all your reads have the same properties. If you've had two different sets of paired reads, that is with different spacing, this is a good time to separate those libraries and record that information in each one, capture that, and pass it on. But again, you know, if all you're going to use this application for is to, or this set of applications, is to run FastQC and then run Trim-O-Matic for trimming, then you don't need to separate all your files. But you still need to fill in all the boxes. You need to, that come, and they may be annoying, but there are a series of boxes that, that give you a chance to better define your files, your library of files. And it also, if you're doing the tuxedo pipeline, the condition or naming box is important because that is the box that defines what kind of samples they are, whether they're wild type or mutant or whatever differentiates them. And an important consideration when you're doing this is that the discovery environment, all the applications run in Linux, and Linux was written starting in the 1960s. A lot of the code was assembled to be used for running mini computers. I think in the late 70s, early 80s. And so at that point, a lot of it became something closer to what became Unix and eventually Linux. But as you can imagine, if this was a mini computer back then you have an idea that how you used it was a little different, and it was by command line, and no spaces are allowed. So no spaces ever in names in the discovery environment, and not when you're entering them into these applications, because you'll screw everything up. Guarantee it. So let's go on and talk a little bit about FastQC. FastQC is the first step in the software that's actually the main step that's run by the prepared directories run FastQC step, as FastQC, as the name implies. And it does nice things like puts together graphs and assembles information. It summarizes it. It does all this in a way that can be helpful to you in understanding your data and better assessing it. So let me now go to the discovery environment. And I assume that most of you have seen the discovery environment before, have used it. Let me pull this up a little bit. And here, I wanted to look at some jobs that I ran in the past, and I think many of you, if you have run things in the discovery environment in the past, you probably know that if you select in a job like this one that was run just a few days ago, really, and click the app name, then you open a window, as is open here, and you see that you could rerun this application all over again. You can change the inputs and the settings if you want, or you can just let it run exactly the way it should run to duplicate what happened before. Now, I wanted to just show you how it looks to enter data here. And here, for example, are the inputs. There's where the left read files go in. There's where the right read files, the unpaired reads. And so it assembles that information together. And the data used here is, they're conveniently labeled frag for fragment and short jump for short jumping types of libraries. I guess in this step, we've separated those because they have different spacing, even though there are only 4 files for this whole data set. If you're interested, the frag data, it represents genome reads, genome sequencing for Rhodobacter spheroides, and that data can be found in the discovery environment. But I just wanted to take a slight diversion here and show you. If you have a question about anything, and in this case I can tell you that data came originally from the GAGE project, which has some nice sample data for genome assembly if you're interested in that, or if you just need some read files to play with. And since I've told you it's the GAGE project, I can just type in GAGE project, and voila, it pops up GAGE. And it may do it a little more easily for me because I've done this search a number of times, but still, it's not too hard to find this by just putting information in. And the data sets are listed here, and you can download them. And this should be your general approach when you have questions in bioinformatics about applications in the discovery environment. But you can also find these data by going into the data store, going to the community data section, going to the iPlant training, and reference genomes, I believe. No, actually, in this case, I think it's easier just to go to the all-paths LG because it's data that you can use for this particular assembly. And so here is that data. FastQ files for frag, FastQ files for short jump if you want to test those too. But here, those files are already entered in, ready to run. The other thing that we need to look at then is we have all these boxes, as I said, to fill in. But these are just names. The main thing you have to avoid is no spaces. But this is your chance to name your library and make use of that information. And here you should enter the paired end spacing and the standard deviation on the spacing. And also answer a simple question, are they fragment type reads or jumping type reads? That has to do with the type of pairing. And if they're not paired, then just leave it fragment and otherwise ignore it. And if you're doing nothing with the HTProcess pipeline for now, except running this application and the one that follows, the HTProcess Trimmomatic application, then all you have to do is enter a number here, just make something up. But for the fragment reads, the spacing is 180. And just a guess, because normally you pretty much have to guess what the standard deviation is going to be. And typically that's going to be, let's say, 20%. And so I stuck 40 in there. And then you can launch the analyses. And once that's run, you can get results like these. And it'll take a minute to open this. And it may not take a minute for you to do this, but it does for me because I have a lot of files, because I run a lot of things through the discovery environment. But while it opens...
While you're waiting,
yes.
Roger, I want to test something. Ramona, could you unmute and speak and see if Roger hears you?
Can you hear me?
Do you have your...
I'm speaking now and you can't hear me, right?
I have my microphone.
Yeah.
But you mean the audio?
Yeah, I'm talking. You can't hear me. So.
Okay.
Now.
There we go. Okay. I'll mute myself again.
Go ahead, Roger.
Okay. Yes, I didn't mean to shut you all out, but too many voices could be a little confusing. And after you've run this job, this is what the results look like. This is not important, but it gives you some files that match up to your paired reads. But there are some folders that match up to your paired reads. It gives you the very important HTProcess reads directory, which contains your read files, which you might ask, well, why didn't I just put them in that box and say we're done in the first place? Part of it is that we need to record the information as it goes in. And this is the most fail-proof way of doing that I could figure out. And a manifest file is created, as I said, to help record information and pass that on to other applications. And it's just recorded in simple text format. And so you can check it, but it's also there for you as you go through the analyses to keep track of your data and what's been done to it and what the different files are. So it's nice to have this as a record. You also have a log file, simple log that just tells you what happened and when it happened. And otherwise, it packages together your reads in one place, so it's ready to be moved into the next application just as this directory without doing any, putting anything else in often. Although, if you're doing mapping, for example, you need reference files. But as far as your reads and information, it's all packaged right here. Your information related to FastQC, on the other hand, is summarized here. And this is a handy way of presenting the data so that you don't have to dig through a lot of individual directories or try to download each set of data separately and in order to open it and read it. You can just click it and open it as I just did here in the discovery environment. And there you can see a summary for the FRAG1 FastQ file and also for FRAG2 FastQ file. And you see information produced by FastQC summarized in a tabular format. And for example, here you see the per base sequence quality was not good for frag1. Well, you click that and see here further down on the same page is information about that file. And here it summarizes a lot of the information that has also gone into your manifest file to some extent. For example, the encoding, that is the FastQ formatting. But you also see here that the quality of the read as you proceed along the length of the read, that as the sequencing process went along, as is often the case, the quality became less consistent across a lot of the individual reads in the file. And so this helps give you information in deciding, well, how am I going to clean up these reads in the next step? But you also have information on the sequence content, how consistent it is across the reads. You hope for consistency. The GC content, duplication levels, and after a while you get sort of used to seeing these things, and you see the Kamer content seems to not be awfully consistent, but across the reads, suggesting that there may be a lot of duplication in the reads. Duplication of some of the same sequences over and over again. But considering that we're looking at Rotobacter data with next-gen sequencing, which tends to give you pretty high coverage, then that may give you an answer as to why you're seeing so many repeating sequences popping up, because some of those sequences are probably showing up fairly heavily represented in the read files. And so one of the other things I might point out that at this point is, you know, as I said, in the next step we'll be doing trimming. Since since these data include the information on the quality and its decay over time, then you may realize that there's considerable loss of quality along here, and you want to do quality trimming, and you almost always want to do quality trimming, but you need to decide where you want to place the cutoff. And how do you do that? Well, it depends, one, on how much data you have, and you may look at the amount of data that you have for this and the overall coverage for bacteria, and you may say, well, in this case, I can afford to throw away a lot of my reads or a lot of data within the reads because I have still so many reads. It's likely that I'll have plenty of coverage for what I'm doing, and I don't want a lot of errors unnecessarily. On the other hand, you might be looking in some cases at saying, well, this is, if this were transcriptome data, well, I'm not going to get very good coverage for low expressing genes, and that's something that's very important to me in my project. So you want to be maybe a little more lenient in how you choose your cutoff. But one way or the other, you need to choose a cutoff point for your reads. In some cases, you may separate where you cut off on this end versus this end, depending on which trimmer you choose, because the next step will give you a selection of quality trimmers. But in any case, for this end, which is often the most important end to consider as far as trimming goes, you see, you may want to cut off, if you want to save a lot of reads, then maybe you want to cut it off around 15. If you feel you have plenty of reads, then you may want to cut it off at 20. But this is where your judgment comes in. You have to decide for your project. And there may not be any one perfect answer, and you don't necessarily have to stick with your answer. If you find after running the HTProcess trim-o-matic step that follows, maybe you'll decide, well, I didn't like the results there, so go back and do it again, starting with the results from this app. So that's how you approach things overall. And so at this step, or at this point, maybe we should go back, take a look at the next step along the way in the pipeline, the HTProcess trim-o-matic step. Yeah, I think this may also be a good time to take some questions. If you have questions related to what I've said so far, if you do, probably the easiest thing to do is just type it in the chat window.
Since we only have 5, they could also unmute and speak, probably. Up to you, though.
Okay.
That's fine.
Yeah, well, if you prefer, you can just unmute and speak too.
Yeah, you're going to give a prize, right? A prize to anyone who asks questions?
Well, I'm going to give them some credit right now and say, okay, this stuff, running FastQC is pretty straightforward. And as I hinted before, if you really have a question about FastQC and how it works, the best way to find out is just by putting sequencing FastQC in Google and looking for the manual for FastQC. So that's what I do when I have questions. And a lot of the time, otherwise, I'm guessing, just as you might, because sometimes the manuals themselves are sort of sketchy. So you have to get used to interpreting them. But that's where I can help you in some cases. You might feel that I'm better at interpreting them. Sometimes I'm just braver in how I guess. And that's what it comes down to. And as I said, if you're not sure, then try things and see how it works. And if you don't like the results, go back and run things again. And there isn't necessarily one way of running everything. When you're running assemblies, for example, your Kamer settings can affect things drastically, and yet even when you do your best in choosing a Kamer first off, you may find that adjusting that Kamer setting or just trying different settings may lead you to a better result. So a lot of this is based on judgment acquired through experience, but also through willingness to test things. So don't be too afraid. So the next step, as I said, is HTProcess trim-o-matic. And as I hinted before, when you use this pipeline, all you need to do really is to drag the HTProcess reads directory, HTProcess reads directory from its location in the data window into the appropriate box for the HTProcess trim-o-matic app. I guess you can also access it by browsing, but I usually just do drag and drop because it's easy. Then you have to decide how you're going to set up the various trimming functions. And there are a lot of trimming options with HTProcess Trim-O-Matic. Trim-O-Matic is the application, and the reason I chose it is because it is so flexible. It's like the Swiss Army knife of trimmers. And it is a paired reed trimmer, so it doesn't break your parent. This is another question that often comes up in the discovery environment. When you're doing trimming and you have paired reeds, use a paired reed trimmer. And Trim-O-Matic is not the only one. Sickle is another one, for example. But if you don't use a paired reed file compatible trimmer with your paired reeds, and it's usually, I mean this specifically for quality trimming, then you could break the pairing, and that will screw things up entirely. And you don't want to break your pairing because you presumably paid extra money for having paired reeds, and you get a lot of good information from that pairing. I guess that's the end there. So let's go back to the discovery environment. And take a look at Trim-O-Matic step. Let's take a look at how we enter data into it. So this was how I ran this step, and this, I believe, yeah, was for the same set of data that we were just looking at, the frag1, frag2 reads. And so here I entered the HTProcess reads directory. And then we have all these settings for different trimmers. Do I need to use all the different trimmers? No, you don't. Pick the ones that you want to use. If for some reason you feel that your reeds need no trimming whatsoever, you can probably enter absolutely nothing, but I'm not sure that that would work. But usually it's a good idea to set at least some quality threshold and put it through, just in case there are a few odd reads in there that are really bad quality but aren't showing up in the graph on FastQC. So in this case, I chose to use the ImmunoClip program, and you may almost always want to use the ImmunoClip, the IlluminaClip trimmer, because that one helps remove extraneous sequences that come from the sequencing process itself. It helps remove any Illumina adapters or primers that are left on your sequences. And so the question is, what do you use often for a file for this? Well, as you can see here under iPlant training, there is a read cleanup tutorial for separate from this, but we can still use some of the files, and one of the files, the input files, is Illumina adapters in FASTA format. And so you may want to use this file. It's just an example. You may want to pick out your own set of adapters that you know that your core facility was using. You may want to throw in some of your own primer sequences if there were any primers used in cloning or amplification along the line that may have contaminated the reads that we're going to be working with. And then otherwise, for the Illumina clip trimmer, I usually go with pretty much the defaults. And defaults usually can be found by just mousing over the little eye that's at the end of the boxes. And in this case, default is 2. It gives you a range for this setting. And this palindrome setting is, it's a little complicated. I recommend looking it up in the Trim-O-Matic manual. And I assume you know at this point how to find the Trim-O-Matic manual. Hint, it's on the web someplace. Same with this setting. This is just for simple clipping, but it gives you, I think, an amount of overlap with the adapter sequence necessary before you start thinking about clipping off this sequence. Now, beside this Illumina clip trimmer, there's head crop, which trims the front of the read file or the read itself, and you can choose a fixed value to trim off, and I can show you an example when you may want something like this later. You can also use crop to do the same towards the end, but the difference is up here for head crop, you enter in how much you want to take off. For crop, you enter the value of what you want to leave behind. And then here, it's often good if you're doing quality trimming to enter a value for what you want to leave as a minimum read length. And the thinking behind this to some extent is, well, if I'm going to have to cut off, say, 40% of a read, maybe the rest of it is sort of doubtful too. Maybe it just wasn't a good read at all. But you have to use some judgment and decide again, how much do you need that read, and how likely is it that it's going to be all bad? Or also how useful it may be for your downstream applications if it's too short. That's another consideration. Now for quality trimming, you have choice of the leading and trailing ends, and you can set fixed quality values for where you do the cutoff, and that's what we were talking about before. Leading, in this case, I chose 18, and trailing, I chose 15. Very, leaning towards saving a lot of the reads. The sliding window method, I assume on the name, and you can do your own reading on this, chooses a window size, and you see here the default is 4. So when I use this one, I often use 4, and then you just choose the average quality. It's not the quality of where it drops down to a certain threshold for that specific base. It's when it's centered at a certain base, the average quality goes below a certain value. And then there's the max info method, and that's a little more complicated, but it's designed. It uses some relative numbers to decide based on how important it is for you to retain information or your willingness to lose it, that is read length, again, read information. That helps you, it helps decide whether to do the trimming in a more conservative or less conservative manner along the way. So it uses a more loose set of decision making, and that may be good, but I'm not as comfortable in using that. I just haven't used it as much. And so if, in most cases, these boxes represent all you need to fill in for running Trim-O-Matic. But you'll notice down here, there are a whole different set of boxes. And that's for program 2, program 1, program 2. What is that about? Well, let's go back to, well, let's open this one up for now. If you look at the manifest file, after running the HTProcess prepare directories, run FastQC, to see that the trimming step is set up by either or both paired reads and single reads that are set up for trimming. And you can pick those out pretty easily by the word trim in the name here. Essentially, this is a flag to tell the next program, this is a read to be trimmed. And this one here tells it to use program 1. If for some reason you want some of your reads in this library to be trimmed slightly differently, you could set this to a 2 and enter information in program 2. And how would I do that? Well, you can literally go in here and edit this. And that's how this whole app was designed, to give you that much more flexibility. And most of the time, since you're dealing with a library of reads, you won't necessarily need this level of granularity, looking at each read file separately. But in case you do, this does give you some flexibility. And the other thing I wanted to point out here in this particular set of reads, and you may notice that this was a different set of reads. This is some of my own data from a number of years ago. And I'm showing you my data just so that you understand. We all have problems with our data, and you can see I had problems with these data. And you sometimes see odd things like this, where the quality drops out at one step along the sequencing process. That it doesn't destroy the value of the reads necessarily, but you do have to consider what kind of trimmer you're going to use. Take care of that. Another problem that I see in these data, and that you will see periodically, not just in my data, I've seen it in other people's data fairly frequently, and you see all this happening. Instead of fairly flat distribution of base sequence content or GC content, you see this jumble of peaks, suggesting, as far as I can tell, that there are some repeated sequences showing up there. And what kind of repeated sequences may be showing up on so many of the reads? Well, my guess is that they're adapter sequences or primer sequences. And when I ran Trim-O-Matic the first time with this file, for example, I found that just using quality trimming didn't get rid of this. And so I was left with the decision, well, do I leave that in and just assume that since it's not a big percentage or a huge percentage of the reads that it's not going to screw things up too much, or do I want to get rid of that? And I decided that I wanted to get rid of that, and that's where I used head crop to just take that off the beginning of the reads. You know, and there's a cost there, but the benefit is I feel more confident that I'm not trying to assemble sequences out of junk, and that's what it comes down to. You know, these experiments are our children in a way, and we hate to be harsh with them, but sometimes we have to be. And so I think that's most of what I need to say about these two applications and describing how to use them. We have, again, the HTProcess, prepare reads, and run FastQC, and we have Trim-O-Matic, the HTProcess Trim-O-Matic. And if that's all you ever use out of the HTProcess pipeline, I think you will find that it can serve you well, especially because it helps you keep your data in an intelligent collection form and also gives you this essential feedback. And I guess one of the things I didn't show you, but you would probably be able to figure out on your own, is that when you're looking at the results for Trim-O-Matic, it also has a FastQC Summary file. And this is a different one because after trimming, this application reruns the FastQC set so to give you immediate feedback as to how well did you do in trimming. And this is the same set of data, the FRAG1, FRAG2, but in this case, I included FRAG1 and short jump 1 and short jump 2 all together in this library, even though they didn't share all the same properties, like pairing distance. But I did this to show you that if all you're going to use it for is for running these two steps, then you don't necessarily have to separate the data. But you can also see that after trimming, we see that the quality is much improved for these files. This is FRAG1, which we were looking at before. And instead of sagging down here, we have a fairly reasonable set of reads and read qualities that are dipping down only to 20 in this case. A little further for short jump, because I think I actually used the second program for this because I found with short jump that we had fewer reads after doing trimming like this. We just didn't have nearly as many reads, and I decided, okay, I'm going to give short jump reads a break. And so I trimmed them less harshly, and that's why you see the error bars coming down closer to 16, which I think is what I used instead of 20. So that's where that second program can come in handy, especially if you're using a mixed set of libraries, which under some circumstances I may yell at you for doing. But in this case, if you know what you're doing, it's okay. And another thing to notice is that sometimes you'll see overrepresented sequences coming up in this information that's put out by FastQC. Sometimes you may want to go in and it's interesting to blast it. Just go to NCBI and blast this sequence and say, what the hell is this? And you might learn something that you're sad to learn about your data, that maybe this is a contaminant, or maybe this is from a totally different species, and like your own. Maybe it's human sequence data mixed in with your bacterial sequencing project. It happens, and so you may want to address this, or if it represents a very small percentage of your reads, then you might say, well, okay, I can live with that. But there are also programs that can be used to remove these contaminants, and FastQ Screen, for example, will help you with that. But that's a totally different application and a different talk, I guess. So how about we open it up for questions at this point? I assume that some of you understand completely at this point, but some of you may have questions. You may have questions stemming from your own experience with your own data in trying to do trimming or quality analysis. And you may want to try to understand better what to do under different circumstances.
Are there any questions either in the chat window, or you can unmute yourself?
Let's see. Yes. Uwe is pointing out that there is another read cleanup and pre-processing tutorial. And that's one where you can use, if you're dealing with a small number of read files and you want to just do it one file at a time, you can use, for example, sickle and scythe, which are mentioned down here. Sickle is a paired read quality trimming program, and so it's a good choice other than Trim-O-Matic for doing trimming of paired reads. Scythe is one that is sort of like a Lumina clip, where you're going to be clipping off specific sequences related to adapters, for example. And you also notice here, I point out something, most of the time we think of getting rid of the bad data, and erroring as much as possible towards having totally clean reads. But under some circumstances, you'll get better results if you're very sloppy, let's say, or err towards the dirty side of things when it comes to quality trimming. And that's because sometimes you lose depth of coverage, or you lose what may be a hard to cross stretch of sequence that may end up looking like it has lower quality, but it's an important stretch of sequence within a genome, for example. And bridging that, it may be important for extending the size of your assemblies. And you want to preserve that information as much as possible, and because it can be recovered to some extent if a good error correction program may be used to correct where just random noise is coming in, and salvage the good information that's still there. And that's, to some extent, requires the judgment call of a good quality evaluation error correcting program. But AllPads LG, the assembler, actually uses error correction. And so that's one application where I suggest don't be too harsh about trimming, because I've found that you can get better results by not trimming too harshly. And
again, are there any questions?
Any questions? I don't see any questions popping up here.
Okay.
I assume you're all comatose or you totally understand what's going on at this point.
Okay, so I'll squeeze in here. I'm Martha. We want to thank everyone for attending. In the chat window, I put a link to a survey so that we can get some feedback from you all. These topical webinars that iPlant has started doing are new. We need your input so that we can make them useful to you. Yeah, last call for questions, anybody?
Yes, the questions generally about bioinformatics, iPlant, the meaning of life.
Yeah, post them on the Ask forum. I put the link to the Ask forum in the chat window also.
Yes, and we always take questions, and we usually answer them in the Ask forum for iPlant. And you often see me popping up there trying to help.
Okay.
Okay, well, thanks for attending. So you know where to find us.
Okay, and I will send a follow-up email with the links to the recording of this and the survey link. Thanks, everybody.
Thank you. Bye-bye.
Thank you, Roger.